Hello, welcome to our 2019 Science and Discovery webinar series. My name is Emily Taylor. I'm the Director of Advocacy and Community Relations for the Solve MECFS initiative, ME being myalgic encephalomyelitis and CFS being chronic fatigue syndrome. We are so lucky today to have Dr. Amy Proal, presenting on MECFS in the era of the human microbiome. Persistent pathogens drive chronic symptoms by interfering with host metabolism, gene expression, and immunity. This is part of a webinar series on a series of publications from Frontiers in Medicine, all about MECFS. And so we've been exploring each of these papers and the impact they have. And we're really excited to have Dr. Amy Proal, who authored one of those papers, today. She'll be speaking about that particular article. In addition to, we'll talk a little bit more in depth about, she's a recent Ramsey Award winner as well. So with that, just a few facts about Amy. Amy Perl PhD is a microbiologist who studies the molecular mechanisms by which bacterial, fungal, and viral pathogens dysregulate human gene expression, immunity, and metabolism. Her work further examines how dysbiosis of the human microbiome and the human virome, which means viruses, can contribute to the chronic inflammatory disease process. As a member of the research team at the Autoimmunity Research Foundation, Dr. Proal has authored papers and written book chapters for organizations like the J. Craig Venter Institute and the Autoimmunity Network, and lectured at the NIH in numerous international and US-based conferences. Dr. Proal, we're so lucky to have you today, and with that, we'll take it away.
Hi, thanks so much for hosting me. So I'm excited to talk about this topic today. I'm going to talk a little bit quickly because there's a lot of content to get through, but my goal is that you can go back to my slides and kind of fill in the gaps. So yeah, I'm going to talk today about how persistent pathogens can contribute to the illness myalgic encephalomyelitis chronic fatigue syndrome, which is MECFS for short. Hold on, let me just, I need to be able to move my slide forward better. Oh, hold on. Okay, there we go. Oops. So millions of patients across the globe are suffering from this condition, MECFS, and currently there is no one specific disease biomarker, which means patients are diagnosed off clusters of symptoms. And when patients are sick with this illness, they become very sick. In fact, severely ill patients are often wheelchair dependent, bedridden, and unable to perform basic tasks of work or daily living. So myalgic encephalomyelitis chronic fatigue syndrome. Why are there two names? Myalgic encephalomyelitis is derived more from the United Kingdom. Chronic fatigue syndrome is more the US name that this disease got. I personally like myalgic encephalomyelitis. It refers to swelling of the brain. I think that's a pretty okay term. I really do not like the name chronic fatigue syndrome because fatigue implies that people are just a little bit tired, when really this website, this is just the CDC listing some of the debilitating symptoms that people with this condition actually have. So beyond fatigue. So we have muscle pain and sore throats and sensitivity to light and sound and completely unrelenting fatigue. And in fact, also a symptom called post-exertional malaise, which is an incomplete ability. Patients cannot recover from exercise at all, even small, tiny bits of exercise. So, beyond that, MECFS may be a neuroinflammatory condition. It's increasingly being studied in that capacity. So for example, here, this is a study by researchers in Japan, and they found increased microglial activation in the brainstem of MECFS patients relative to healthy subjects. And that is an indicator of neuroinflammation. Jared Younger and team at University of Alabama are using different techniques to look at neuroinflammation in the MECFS brain, and so far these tools have shown some signs of neuroinflammation. And Mike Van Elzeker's team, who's working on this at Harvard, is doing brain scans on patients with the disease too, and his early data is also leaning in the direction of a neuroinflammatory type condition. So what about us? As was mentioned, our team wrote a recent paper that looks at persistent infection and what's called microbiome and virome imbalance in this condition, and how does that tie into neuroinflammation and other parts of the disease process? Well, For one thing, let's just get to this point. Why would we study persistent infection in MECFS? And one of the reasons is if you study MECFS through the lens of persistent infection, the disease is far from a mystery. Sometimes this illness is referred to as mysterious, and I don't think that's the case. For example, you know, there have been multiple outbreaks of MECFS, of these clusters of symptoms across the globe. In fact, you can see here, this is a 1956 article from The Lancet, and already there are several outbreaks listed on the right side that have occurred in many different places. And, you know, it seems very clear that a pathogen was at least involved in these cases. In some of the outbreaks, the pathogen was actually identified, for example, a Coxsackie virus or a poliovirus. And in other cases, the pathogen wasn't identified, but then again, the tools that were used or available at the time to identify viruses and other pathogens were not that great. So again, beyond that, also, MECFS, you know, even cases that are not tied to an outbreak, most patients present with symptoms after suffering from a severe bacterial, viral, or even fungal infection. And these infections often correlate with travel to a foreign country or exposure to pollutants or molds, which suggests that some of these pathogens can take advantage of factors that compromise the person's immune system. And in fact, this has happened so often with these patients that at different points in history, MECFS has been called post-polio syndrome, chronic mononucleosis syndrome, or post-viral syndrome, due to the fact that chronic symptoms are so often noted in these patients after acute infection with pathogens like Epstein Barr, the enteroviruses, influenza, or a range of other pathogens. And then patients' symptoms are often very flu-like in nature. They suffer from sore throats, often swollen lymph nodes, and these symptoms tend to relapse and remit as would be expected if an organism or a pathogen was involved in the disease. And beyond that, there's been cytokine activation and immune cell activation documented in the disease, and that kind of inflammatory response is often driven by infectious agents. So also, you know, there's a big trend out there. There are a growing number of what are being referred to as post-infectious chronic conditions. So if you look up any pathogen right now that's, you know, a big problem in our world, Zika, measles, Ebola, influenza, dengue, polio, Borrelia, beyond that, there are cases in which people who are exposed to these pathogens in sort of an acute form, some of those patients go on to develop chronic symptoms based on that infection that do not go away. It's a chronic form of that condition. And what's really important to understand is that in many of these cases, the infecting organism can still be detected months or years after initial infection in certain tissues of these patients. So, for example, in some of the post-Ebola cases, there was you couldn't find the Ebola. Research teams couldn't find it in the blood anymore, but 2 teams were able to find it in patients' eyes, and 1 team still found it in men's semen years after initial infection. Oops, I don't know how that happened. Hold on. So this trend is already played out in MECFS. So John Chia, who's a doctor in Southern California, he did a study in which patients who were admitted to a hospital for acute enterovirus infection, he tracked them, and 3 of the patients developed MECFS symptoms. And so he biopsied got stomach samples from these patients, and he did find the persistent enterovirus in those patients' stomach biopsies. And so, as you'll see, I'm not arguing that an enterovirus is driving every single case of MECFS, but this is a great example of how a virus, a chronic virus, can drive the symptoms associated with this disease. So beyond that, there's another important trend. If MECFS is a neuroinflammatory condition, well, we should look at what other communities also studying neuroinflammatory disease are doing. And one of the biggest trends in the Alzheimer's community, the Parkinson's community, ALS, MS, is growing evidence that pathogens able to persist in the central nervous system or in the brain are contributing to these diseases. And so here's a few studies I put up as examples. The study on the left was done by a team at Mount Sinai in New York, and they found a range of herpesviruses in the Alzheimer's brain. The study next to that one is a different team that found a very serious bacterial pathogen called P. gingivalis in the Alzheimer's brain as well, and even the proteins that this pathogen creates in order to drive disease in the brains. The study on the left is another team from Spain who does great work, and they found a range of bacterial and neural tissue from patients with ALS. And here's an example of research in Parkinson's. This team found enterovirus antigen and virus-like particles in the brainstem of people with this condition. And remember, enterovirus is the same virus that Chia found in the stomach of patients with MECFS. And so what you can already see there is that these viruses and pathogens can infect different body sites in different patients or in different conditions. And so there's another major breakthrough that I think people should be aware of in the neuroinflammatory community. And in that picture, you can see Rudy Tanzi and Robert Moyer, and they're researchers at Harvard that are actually advisors on our team's current Ramsey project. And they have found over the past couple of years, it's a huge breakthrough. The plaque that accumulates in the Alzheimer's brain, their research shows that it's not just some useless plaque. It's actually a potent antimicrobial peptide. And what an antimicrobial peptide is an important part of the human immune response towards infectious agents in brain tissue or other tissue. And so the study on the left is one of their first studies on this topic. They showed in the lab that amyloid beta, this so-called plaque, was rapidly seeded in response to herpes viruses. And then they did a second study, which also showed that amyloid was, induced formation of amyloid happened in response to bacterial pathogens like salmonella or fungal pathogens like Candida albicans. And so this is their running model for the disease, which I took from one of their papers. And so basically pathogens enter the brain, then amyloid, this part of the immune response, forms around that pathogen. And in simple terms, it begins to digest the pathogen in H2O2 or bleach. And then that kind of becomes a plaque, that pathogen amyloid complex. And that plaque can accumulate in the brain, but the important thing, it was originally created in response to a pathogen. So a takeaway from that is that the presence of amyloid in a sample could indicate the presence of specific pathogens. And that's something that we're following up with their research team that maybe we could apply to MECFS. So beyond that, it's not just their team. By the way, there's other teams finding similar trend with amyloid. And in Parkinson's, there's a similar protein called alpha-S-nuclein that's associated with the disease process. And different teams are finding that alpha-S-nuclein also appears to be an antimicrobial peptide that forms in response to pathogens in patients with Parkinson's disease. So this is a growing trend in the neuroinflammatory disease community. So, why am I comparing MECFS to conditions like Alzheimer's disease? Well, I think there's a lot of comparisons that can be made. And for one thing, the cognitive impairment that many MECFS patients experience can be extremely debilitating. This is information taken from a presentation I gave a few years ago. And if you look at the first woman's comment, she says, I went from being a company executive to barely reading over the 4th grade level when she got sick. And that's common. That is normal for patients with this illness. Beyond that, there's one research team that did an autopsy of an MECFS brain, and the brain was full of amyloid, these same so-called plaques that the Alzheimer's community is now researching increasingly as antimicrobial peptides. And I showed this study to Robert Moyer, who's on that team, and he was really interested in it. It's the same kind of amyloid formation they're seeing in Alzheimer's. And a team in Georgetown a while ago also did a study that looked at the cerebral spinal fluid of patients with MECFS, and they found amyloid proteins in there as well, and also other innate immune system proteins. And they even replicated that finding in 2 different cohorts of patients with a disease. So, that could indicate the presence of organisms in the cerebral spinal fluid. So beyond that, in the brain, there can be formation of what are called virtual Robin perivascular spaces. And these spaces are kind of spaces that surround blood vessels in the brain, and they're associated with increased inflammation. And the more of these spaces the brain has, the kind of, and generally, the more sick the brain is, in simple terms. And these virtual Robin perivascular spaces are common in the Alzheimer's brain, but this is unpublished data from Mike Van Alziker's lab at Harvard, and his team is finding these same perivascular spaces in many of the MECFS brains that they're analyzing, and this is early unpublished data, but that's an interesting trend. So beyond that, this is a really interesting study done by a pathology team at Harvard that was published just about 2 weeks ago. And they were looking at autopsied autistic brains taken from patients with autism. And they found these same perivascular spaces in the autism brains as well. But in this case, they were able to look further, and they found that these spaces were surrounded by immune cells called astrocytes. And blebs of sort of kind of toxic material were coming out of the astrocytes, and those in turn were surrounded by what are called cytotoxic T cells, which are T cells that target pathogens. So Matthew Anderson, who is actually at Beth Israel in New York, who was part of the study, he said in a press release on the study, either the T cells are reacting normally to a pathogen, such as a virus, or they're reacting abnormally, for example, like an autoimmune disorder. And I would argue that the first trend is more likely. So another major trend when we're looking at pathogens, organisms that play a role in MECFS, we have to look at what's called the human microbiome. And that's because our bodies are filled with vast microbial and viral ecosystems. These are communities of organisms that interact. And many people, I think, are familiar with the gut microbiome, these vast communities of organisms in the gut that contribute to many of our human processes, the way we digest food and the way we break down different compounds. But it turns out that over the last couple of years, more research teams have been using tools to identify organisms beyond the gut in the human body. And these microbiome ecosystems of interacting organisms extend into nearly all human tissue and blood. So there's a microbiome in the skin, in the lung, in the nose, in the bladder, in the pancreas, in the liver, in the blood, even in atherosclerotic plaque. And, of course, we're looking more at the central nervous system, and no longer seems sterile either. So, again, what kind of organisms are in these communities? We have bacteria that, are the most kind of well-known organisms in these communities, but fungi are also there. There are DNA and RNA viruses. There are archaea, which are organisms that are kind of like bacteria, but a bit different. And there are also different forms of viruses, especially viruses that are called bacteriophages. And bacteriophages are viruses that infect bacteria. And when they do that, they modulate the activity of those bacteria and completely change the way the bacteria can behave. So it's almost like a bit of a Russian doll model going on here, where you have interacting organisms, but often they infect and modulate each other as well. So moving forward from there, all of us have these microbiome communities in us. An Olympian, a champion, anyone climbing a mountain has all microbiome environment ecosystems, and they're in a state of excellent health. So obviously these communities can persist in and on us, and people can be healthy, but it's really important to note that the microbiome is not always good. These communities can shift their activity, and I'll explain that a bit more later, in ways that can also drive disease processes. And the study on this slide is a good example of that. There now appears to be a tumor microbiome. This is basically communities of interacting organisms in tumors, where the pathogens are usually inside the cells of the tumor. So this was a study that looked at pancreatic cancer tumors. And believe me, there are a lot of pathogens inside those tumors, and these pathogens are basically driving a lot of the gene expression changes that progress the cancer forward. So, beyond that, it's important to note that we're just in the middle, the research community, we're just in this middle stage of even identifying and characterizing these organisms that we now realize are capable of persisting in the human body. And this is a good example of that. This was a study done just about 2 years ago by Stephen Quake, who's a researcher at Stanford. And in one study, he used a new technology to look for bacteria and actually viruses as well in the blood of immunocompromised patients. In that study, he found thousands of new organisms in the blood of these patients that had never before even been understood to be able to persist in the human body before that study was done. So on the right, you can see a press release associated with that, and it says Stanford study indicates that more than 99% of the microbes inside us are unknown to science. So yeah, we're still working on even understanding what organisms are in us that might have the capacity to drive disease. And in fact, Quake at the end of that paper concludes, quote, these novel microbes have potential consequences for human health. They may prove to be the cause of acute or chronic diseases that to date have unknown etiology. Okay, that seems, let's move from that. So, a lot of teams just don't look for organisms in the blood. It's a newer trend, but if ye seek, ye shall find. This is an example of a study done by a team at UCLA, and they looked at bacteria in the blood of patients with ALS, with bipolar disorder, and with schizophrenia, as well as just in the blood of healthy control subjects. And you can see in that figure on the right, there were so many bacteria found in the blood of these patients. And in fact, the diversity and kind of composition of these bacterial communities differs between, healthy subjects and patients with the disease states, especially in schizophrenia. There was way more diversity of bacteria in the blood, and we don't know why yet, but we should certainly try to find out. So, in MECFS, not too many teams have done extensive analyses of organisms in blood, but this study really interests me. This was a team that looked at certain bacterial phyla in the blood. And what they did was they did this after an exercise challenge. So they had patients with the disease exercise, and since patients can't exercise with MECFS, it's basically an exercise challenge is a way of provoking symptoms in such patients. And after this symptom provocation, the difference in relative abundance of 6 out of 9 of the major bacterial phyla in the blood that they were studying increased from the baseline exercise challenge to up to 72 hours after exercise. And that's a really interesting trend. What are they doing? And that's something that our team hopes to follow up on as well. So here is another paradigm shift. This is me in a picture with Robert Moyer and Rudy Tanzi, again, that same team at Harvard that was looking at amyloid. And they're also running what's called the Harvard University Brain Microbiome Project. And what happened there is they were studying, they were looking originally for single pathogens in autopsied Alzheimer's brains, but when they started to really use advanced tools to look for organisms in the brains, they found that all of the brains just had many organisms in them. Even the healthy brains that were supposed to be control brains had organisms in them. So they started this project, which is an ongoing project. The data has not yet been published, in order to just begin to characterize organisms, communities that seem to be in these brains. And if that plays out, then there technically could be a microbiome in the brain. And they're not the only team finding this. This is a team at University of Alabama, Birmingham. They did a separate study that found bacteria in the brain as well. And here's a quote from one of the researchers who did that study. She says, quote, "We saw that bacteria like to be inside astrocyte cells around the blood-brain barrier. They also like to be in axons, which are projections between brain regions that conduct information." So that's interesting, especially because astrocytes, if you remember, were the cells in that autism brain study with the perivascular spaces. The spaces were surrounded by astrocytes, and astrocytes and actually microglia are easily infected cell types in the brain. So we see just a growing trend towards the fact that these cells may be infected in people. So beyond that, you have what's called the human virome. So as I mentioned, the microbiome, these communities of organisms in the human body have different organisms in them. And if you just want to refer to the viral part of those communities, it's called the human virome. And these are vast communities of viruses in the human body. And research on the human virome has exploded over just the past few years due to improvements in tools that are able to find known and novel viruses in the human body. And it turns out that viruses are the most abundant life form on the planet and in the human body. And bacteriophages, or phages, as they're called for short, those, I mentioned them before, the viruses that infect bacteria, well, each study on the human virome is turning up more and more phages that we didn't realize also persist in the human body, to the point where it's currently estimated that trillions of phages persist in human tissue and blood at any given moment. And a current general estimate is that the scientific community has identified and characterized about 0.001% of these viruses thus far in the human body. So just so you know what I'm talking about, this is a computer-generated image of a bacteriophage. So, you can pause for a second there and kind of picture trillions of these, pulsing through your tissue and blood. I'm sure they're doing something. And one thing they are doing is interacting with the human immune system as well. So this is a recent Stanford study by a team led by Paul Bolsky. And this actually draws from research that has been done by teams in Russia and other communities before, but he showed in this study that yes, phages interact with the human immune system in addition to infecting bacteria. In fact, he found that phages directly interact with human cells that can impact and modulate the human immune response. And the paper provides examples of how phages can modulate innate immunity via cytokine responses. They can even impact the adaptive immune system via effects on antibody production. So these phages are doing quite a bit. And Bolsky, in a press release on that study, said, "We've long known that you've got up to 10 quadrillion phages in your body, but we just figured whatever they were doing was strictly between them and your commensal bacteria. Now we know that phages can get inside your cells too and make you sick." So that's a trend we need to follow up on, and that's part of why our team, our Ramsey project, is going to characterize phage as part of what we're doing when we're going to look for organisms in MECFS blood and cerebrospinal fluid, and phages are going to be part of what we look for. And in fact, the team on that is a fantastic team in Berkeley led by Nikos Kripidis and David Pazaspino. And they have been running a large project over the past years called Uncovering the Earth's Virome. And this is a project where they're using novel tools, technologies, pipelines, algorithms to better characterize both known and novel viruses in many environments on Earth, including the human body. And so when they first published on this project in Nature in 2016, that single study increased the number of known viruses on Earth by 16-fold. And by 2018, their database, which contains the viruses that they found, had tripled in size. So at this point, I want to just stop and talk about what happens when a typical MECFS patient goes to the doctor. They go to the doctor, and the doctor, if the patient's lucky, will use basically antiquated antibody testing to test the patient for about 5 to 10 well-known viruses, bacterial species, listed a couple of them on this slide. And if the patient doesn't present with sort of obvious antibody responses suggesting that they might harbor these organisms, it's usually concluded that infection does not play a role in the disease. And I hope you can see that there is a huge discrepancy between what the scientific community is now finding about organisms in the human body that are capable of contributing to disease and what is able to be tested for in the doctor's office. And that is a gap that our team is also hoping to change over time with new research. So why, when I talk about single pathogens in MECFS like Epstein Barr and enterovirus, why do I have to bring the microbiome and virome into that discussion? And the reason is because a lot of these diseases connected to organisms in the human body are basically called polymicrobial diseases. They involve different pathogens or different organisms interacting together to drive a disease state. And so what that means is that MECFS-associated pathogens like Epstein Barr, for example, are actually members of complex microbiome communities. So we can learn more about their activity and the ways that they drive disease by studying them as a member of an interacting microbiome ecosystem. It's a little like as if you were studying a person and you studied how they act alone in the country by themselves. And then if you studied them when they, moved into a city like New York City, they would act differently. And so we would want to better understand how that plays out. So again, another reason to study microbiome and virome in the context of these pathogens is that the microbiome community a pathogen enters plays a large role in determining if the pathogen can survive and persist in its host. So I gave an example of that on this slide. The vaginal microbiome, there are communities of organisms in the vagina, actually plays a major role in susceptibility to HIV. So if a woman is exposed to HIV during sexual intercourse, whether the virus actually sticks around and infects that woman has a lot to do with the activity of these other organisms in the vagina because they compete with HIV. They don't want to give up their space. So it depends on the composition of the community whether HIV can stick around. And that's important in MECFS because one of the big questions with MECFS is why do groups of people who are all exposed to the same pathogen, why does some of them go on to develop chronic symptoms? And, I think that the microbiome community that a pathogen enters could at least be one of the factors that helps determine whether those pathogens stick around in certain people and not others. So I want to go back to what's called microbiome environment dysbiosis. I was mentioning before that microbiome communities that are, can promote health in a person can shift their activity and change the way they act in a way that can drive disease. And when they do that, that's referred to generally as microbiome or virome dysbiosis. Dysbiosis refers to imbalance. Essentially, these communities of organisms begin to signal in ways that the entire community becomes imbalanced, and that imbalance contributes to disease. And so at this point, it's worth noting that almost every well-studied human inflammatory disease is already tied to some form of microbiome or virome dysbiosis. And microbiome dysbiosis, happens in these communities outside the gut. The gut dysbiosis is mentioned a lot, but can happen in any microbiome community. So how does dysbiosis happen? Well, one of the main drivers of dysbiosis are dominant pathogens. And these are pathogens, which are kind of like your enteroviruses and Epstein Barrs, whose activity and whose signaling is so profound that it begins to negatively influence the activity of other nearby organisms around it. So dominant pathogens, as a rule, they're big inducers of biofilm formation. They often express proteins called virulence factors, which do a range of things to make people sick. For example, endotoxin is one kind of virulence factor, and I think people are familiar with that. Endotoxins can trigger a range of inflammatory processes. And dominant pathogens also tend to persist inside the cells of the immune system, and when they're inside the cells, they can slow the immune response, and so they become big drivers of disease. And so you could kind of compare a dominant pathogen to the head of a gang of criminals. It's an organism that instigates and influences the other organisms around it to join in and drive disease processes. Another major driver of dysbiosis is immunosuppression, because honestly, the immune response, when it's in good shape, is very good at controlling pathogen activity in the human body. But if the immune response is compromised by medications or chemicals or other environmental factors, then dominant pathogens are much more likely to alter their gene expression or the way they act in a manner that can promote disease. So again, how does this happen? Well, I'm talking about organisms changing their activity. And in order to understand that, I have to bring up the term pathobiont. So pathobiont behavior refers to the fact that most commensal microbes, in fact, all commensal microbes in the human body, these organisms in and on us, can change their gene expression. They can turn certain genes on and off to act as pathogens under conditions of imbalance or immunosuppression. And so here's the paper on this slide that provides just one example of this. This is a team that looked at the bacterial pathogen Staph aureus. And what they found is that about 30% of the healthy human population harbors S. aureus as just a normal member of the nose microbiome. But depending on the activity of other organisms around S. aureus and the signaling and the competitive strategies employed by these neighboring microbes, S. aureus can change its gene expression to drive a range of illnesses, from skin infections to life-threatening diseases like endocarditis and even meningitis. So, what I'm emphasizing there is that these dysbiosis changes are often driven by changes in organism and pathogen activity. And I want to talk about this study quickly as an example of that. So this is a team that looked at oral dysbiosis in the mouth. And they were looking at teeth that progressed towards what's called periodontitis, which is basically cavity formation associated with bone decay. And they looked at teeth that remained healthy and teeth that moved towards periodontitis. The organism communities on the teeth, they looked at both those different communities. And what they found, which is important, is that the teeth, the healthy teeth and the teeth that moved toward periodontitis had fairly similar communities of organisms on them. What changed in the teeth that moved towards periodontitis was the activity of the organisms on the teeth. They began to act differently in periodontitis. And so what happened there is there's a very serious oral pathogen called P. gingivalis. It's a bacterial pathogen. And it's such a serious pathogen that sometimes, even though it's only just 0.1% of a biofilm in the mouth, it's able to impair the immune response so profoundly that it becomes a central player in biofilm growth and development and other signaling, tied to virulence factor expression, for example. And so in the teeth that moved toward periodontitis, this dominant pathogen began to act with a couple other pathogens. And it drove these gene expression changes that changed the pH of the environment, that changed how bacteria could create cell walls, that changed their ability to signal with potassium transport, that changed the way the organisms could move around with what are called cilia. And it even changed the way organisms could respond to oxidative stress or the way they could transport iron. So at the end of the day, these changes in activity of the organisms on the teeth led the teeth to become, to the bone decay and the cavity formation. And what the team concluded is that periodontitis progression is driven by the whole oral microbial community and not just a few select pathogens. In effect, under conditions of increasing inflammation and imbalance, the entire oral community appeared to act together as a pathogen. So I want to go back to this slide I showed you before. P. gingivalis is the same organism that was recently found in the Alzheimer's brain. And this is a study by a different team in the UK that recently found P. gingivalis in Parkinson's blood inside fibrinogen clots. And so if we're finding this organism in different body sites, we have to think about the fact that it might also be acting in concert with the other organisms around it in those areas to drive disease in these disease states. So there's two main takeaways I'd like you to get from this. One, a healthy person and a patient with MECFS could harbor the same pathogen or communities of pathogens, but the organisms may be acting differently in the MECFS patient. This is really important because there have been a lot of times in history where MECFS has been studied and they find a pathogen in patients with MECFS, but then they find the same pathogen in some healthy people too. And then there's a tendency to discard that data as if it doesn't matter anymore. But there's no reason to discard that data because, again, the pathogen might just be acting differently in MECFS or infecting a different cell type or infecting a different tissue. So that's an important trend to keep in mind. And two, it is hard to fully understand the symptoms of a chronic disease without factoring in how a dominant pathogen influences the activity of other organisms in the microbiome or virome community in which it persists. So how do organisms do this when they're in these communities and they're driving disease? Well, what organisms do, or pathogens do, but all organisms do, is create a range of proteins and metabolites. It's part of their life cycles. It's what they do. And these proteins and metabolites are somewhat analogous to just the tools they create that allow them to do things in the body. And so dominant pathogens, this is important to understand, are almost always capable of persisting inside the nucleus or center of human cells. And when they do that, they almost always slow the immune response. There are so many documented ways in which pathogens can slow the immune response once they're inside a human cell. But they also create proteins and metabolites that can interfere with human transcription, translation, and DNA repair processes and the epigenetic environment. So basically, in the center of the cell, in the nucleus, that's where our human DNA is constantly being decoded to make our bodies work correctly. And so transcription is the process by which our DNA is decoded, and translation is the process by which that code is translated into the making of proteins that allow our human signaling pathways to function correctly. Well, these intracellular pathogens and the proteins and metabolites they create, as long as they're in the center of the cell, can directly interfere with nearly every step of these important human DNA processes. And so in fact, proteins created by intracellular pathogens can dysregulate the way human DNA is decoded and able to function in an almost infinite number of ways. So as I'm saying, these pathogen proteins can dysregulate activity of our human signaling pathways. And that's important because these pathways control how our bodies work. These pathways regulate all our human processes, from energy metabolism to immune system activity to how neurons signal, for example. Well, all these human signaling pathways contain what are called receptors on them. You can see that in the figure here. And these receptors recognize human proteins, like you can see in the figure. And when they recognize the correct human protein, the protein and the receptor bind each other and change shape in a way that causes downstream signaling of that pathway to move forward correctly. And that results in a response, you can see in that diagram, and that response is supposed to keep a person in a state of health. Well, it turns out there are only so many sizes and shapes that a protein in the body can have. And what that means is that many pathogens create proteins that are very similar in size and shape to human proteins that are supposed to be regulating our signaling pathways. And that similarity between pathogen and human proteins is often called molecular mimicry. And what that means is that a pathogen protein can sometimes bind into a human receptor, and it can fit when it does that, but because the shape is not totally correct, the signals that that receptor then sends downstream are dysfunctional and can drive a range of symptoms. So let me give you an example of that. This is a team that looked at a viral family called Enteroviridae. And what they found is that these viruses, which can persist in the human gut and in human blood, create proteins that are very similar in size and shape to human insulin. Human insulin is a protein. And these pathogen proteins that they called insulin-like proteins can bind into human insulin receptors. And when they do that, they dysregulate all the downstream signaling of the human insulin pathway in a way that can result in the development of diabetes. So, I don't have time to go into this, but what I want to say is at this point in time, there are examples in the scientific literature of how viral, bacterial, and fungal pathogens can create proteins and metabolites that can dysregulate human signaling pathways that control everything from blood pressure to circulatory issues, to autonomic function, to natural killer cell activity, to lactate accumulation in the blood and brain, to glutamate metabolism, to pain signaling, neuropathy, mast cell activation, mitochondrial activity. In essence, a pathogen protein or metabolite could dysregulate, or has been shown to dysregulate pathways associated with this. And I picked these particular examples because they are all common in MECFS. So in theory, at least, a pathogen and the proteins and metabolites it creates could, drive any of these processes in a patient with MECFS. And if you follow me on Twitter, I just constantly, continually post examples of such studies. So let me move this up one more notch. So pathogen and organism activity, when pathogens dysregulate our signaling pathways, are characterized by a high degree of functional redundancy. And what that means in simple terms is that many different pathogens have evolved to dysregulate similar human pathways in similar ways. And this study is a great example of that. This is a team that looked at the mitochondrial respiratory chain. It's a signaling pathway in mitochondria that basically allows mitochondria to correctly produce energy. And these are different receptors in this figure. Different receptors are complexes on that pathway. And signaling, as I mentioned, has to happen for all those complexes in order for the mitochondria to correctly generate energy. But what the team showed is the different pathogens can dysregulate these different receptor complexes at different parts of the pathway. So E. coli can dysregulate complex 2, but rubella virus can dysregulate complex 4, and rotavirus can dysregulate complex 6. but so can M. tuberculosis. And so the takeaway from that is that if there's a patient suffering from mitochondrial dysfunction that's driven by dysregulation of this pathway, they could harbor different pathogens. One person could have E. coli, one person could have rubella, one person could have HIV, one person could have S. pneumoniae. And as long as those pathogens dysregulated the same pathway, the end result would be fairly similar and similar symptoms would develop. And what that means is that no two patients with MECFS, if infection was involved in something like this, no two patients have to have the same pathogens or mix of pathogens to basically allow a similar illness to occur. Here's just another example of that. This is one study that looked at how different pathogens can degrade collagen. And all the pathogens listed on this slide, from Streptococcus to Listeria to Aspergillus to human papillomaviruses, they all degrade collagen actually with the same mechanism of action. So again, if someone was suffering with a collagen-related issue that could be tied to MECFS or a similar condition, they could harbor any of these pathogens and a similar outcome might happen. And what that means is that in MECFS, a research goal should be to better characterize the signaling pathways and tissues and cell types that redundant pathogens, different pathogens associated with MECFS can dysregulate. And I want to point out that I think one of these areas, I already gave mitochondria as an example because I think that could be a very good focus of what becomes dysregulated by pathogens in MECFS. But I also think that the brainstem is also an area of great interest. And the reason that I think brainstem dysregulation may be involved in the condition is for several reasons. The area postrema in the brainstem, Mike Van Elseker talks about this in his talk sometimes. There's an area there, which is basically a window in the blood-brain barrier. That's a barrier that sometimes impedes immune cells and organisms from getting into the brain, where there's a break in that barrier in the brainstem at that point, which allows immune cells and any pathogens in the immune cells to directly get into that area of the brain. And second, that area of the brain is where the vagus nerve enters and exits. And the vagus nerve is it's such an important nerve in the body. It controls, in simple terms, what's called the sickness behavior response, which is how a lot of times the body just recognizes, when the body recognizes inflammation or an organism, it's the vagus nerve that kind of creates the response to that and relays that to the person. And so this sickness behavior response, in my opinion, can account for a lot of the sort of malaise and flu-like feelings that patients with MECFS have. And if vagus innervates at this area and vagus gets compressed or vagus gets dysregulated or vagus itself becomes infected, then you're going to see a change in vagus activity. There's also been a couple cases of craniosacral instability that in patients with an MECFS diagnosis, and that would also imply that brainstem might be impacted in patients with the disease. It doesn't mean that everyone has to have a structural issue. It just means that the brainstem being dysregulated might be able to contribute to symptoms associated with MECFS. And so, tying this together, MECFS is a spectrum disorder. I just threw up a part of the symptoms associated with this disorder on this slide. And I think everyone knows this. No two patients with disease have the same exact symptoms. No one does. And that doesn't mean that this illness is any less serious than any other illness, because quite frankly, every chronic illness is a spectrum disorder. So even in HIV/AIDS, you have this easily detected virus. But when patients acquire HIV knocks down the human immune response so profoundly that then that patient begins to acquire a range of other co-infections that can be totally different in each patient depending on their environment, what they're exposed to. And so at the end of the day, no two patients with HIV/AIDS ever have the same symptoms either. And they're not expected to harbor the exact same mixes of these additional persistent infections. And so it's really like MECFS being a spectrum disorder that could involve different, organisms in different people would not be much different than what we're seeing in other conditions. And so again, beyond just the infections that can impact patients with ME/CFS, we need to understand that there are environmental things that can also add into the disease process, like mold exposure, chemical exposures, injuries, high levels of stress, immunosuppressive drugs that impede the immune system, and again, rare human genome variants. And that's a topic I don't have time to get into today, but the human genome can predispose towards problems in the way that the body deals with a number of infectious processes. And so that plays into cases as well. So I want to simplify this a bit. And if you were going to look at how ME/CFS might develop based on what I've talked about thus far, our team created a model called successive infection. And what that refers to is, look at that first little snowball on the hill. That might be an initial infection. So pretend that's an Epstein Barr infection. The person develops mono and then never recovers, develops chronic symptoms from the mono. Now they have EBV. EBV can infect B cells and dramatically knock down the immune response when it does that. So now EBV is compromising the immune system. Now that makes it more likely that that same patient might acquire a different pathogen. And that could be, for example, an enterovirus. And maybe that enterovirus, depending on what the person's experiencing, makes it into the brainstem and begins to cause problems in that area of the body. And then maybe that same person sustains a head injury. And now all of a sudden, the inflammation from that injury allows the enterovirus and these other organisms to better proliferate. And now the immune system is knocked down further, and maybe that person is exposed to mold. And maybe they wouldn't have been so sensitive to the mold before, but now the mold and the mycotoxins could become a big issue and also possibly get into the brain. So what happens is that each person's unique ME/CFS symptoms may be a mix of the unique infectious and environmental exposures that that person sustains. And as long as those pathogens or environmental exposures or injuries dysregulate similar signaling pathways, for example, mitochondrial signaling or brainstem function, the same general disease state could develop. And so this is, I don't have time to go through this, but if you want to look back, I'm just reviewing kind of the molecular mechanisms that contribute to the way infectious agents act in this model. And I just want to add one more thing about microbiome in this disease, which is that, ME/CFS is passed in families, especially kind of down the maternal line, the mom from mother to child, and it's assumed often that that means that the disease has to be genetic. And that doesn't have to be the case, because we all acquire our microbiome and virome ecosystems from our parents, especially the mother. So babies in the womb, there were actually 2 studies published in the last month that shows that the infant gut microbiome starts to form in the womb. And that, of course, the microbes that form that are coming directly from the mother. And then there's a breast milk microbiome. So if a mother breastfeeds, she's giving organisms to her child that seed this child with bacteria and viruses and fungi for almost sometimes up to 2 years. And there's other, there's a vaginal microbiome. So the baby's exposed to vaginal microbes during birth. And what that means is that the mother might already harbor pathogens or dysbiotic communities of organisms that they could directly pass to the infant. And that would set the infant up in a way that might allow that successive infectious process to begin, in the womb even, or as soon as the person is born, and it could result in the development of a condition like ME/CFS. And again, the human genome can contribute too, but we can't ignore this part of it as well. So directions for treatment, because isn't that what really matters? Well, the Alzheimer's community is already changing. They're developing new antivirals and antimicrobial medications. And this is a study on the right. It's a description of a team. There's a study at NYU that's actually testing the herpes virus drugs, antivirals, on patients with Alzheimer's. And we know from doctors in the ME/CFS community that those antivirals sometimes help people with ME/CFS. And at this point, the Alzheimer's community is beating us on this when we knew it first. So we should get on board. And second, directions for treatment. I think it's important to understand that if pathogens and infection and environment contribute to this disease, then the last thing we want to do is slow the immune response in patients with immunosuppressive drugs. And that's an increasingly growing understood thing in neurodegenerative disease, This is a paper I really like from a team at the Weizmann Institute in Israel. And it's about just treatment, future treatment directions in neurodegenerative disease. And they say, quote, for decades, it was accepted that the central nervous system is an immune privileged site. This view ascribed the inflammation in chronic neurodegenerative disease to autoimmunity. And as a consequence, attempts were made to treat such conditions with immunosuppressive drugs, all of which failed. And they say further, in conclusion, the development of a therapy that boosts the immune system in a well-controlled way and therefore restores and or activates brain immune communication is an outcome of a general shift toward perception of the central nervous system as a tissue that engages in constant dialogue with peripheral immunity. And such an approach is expected to provide novel treatment modalities in order to harness common immune repair mechanisms to combat Alzheimer's and perhaps other neurodegenerative diseases. So, by the way, a team I worked with previously already created an immunostimulative or supportive treatment for MECFS, and some patients did benefit. You could ask me about that later. And this is a bit about our organism identification project that we were awarded a Ramsey for. I'm not sure, what's my time? Like, moderators, Emily.
Hi, sorry, I had to unmute myself. Yeah, we're actually a little past time.
Okay.
So if we want to get into questions sooner rather than later, but we can start on that area.
Okay, cool. Yeah, so you could ask questions and I can go into the project later, or the project will be kind of described on my slides that people could see later.
Wonderful. Well, are you sure you have no more final thoughts? I don't want to rush you.
Actually, yeah, I want to end with this thought. This is one of my earliest mentors, Gerald Deming. He was a fantastic microbiologist who looked at persistent pathogens in the body in the 1960s and tied those organisms to a range of chronic diseases. And in one of his most famous papers, he wrote, "It is unwise to dismiss the pathogenic capabilities of any microbe in a patient with a mysterious disease. The so-called autoimmune conditions in which no pathogen can be identified by routine testing are particularly suspect." And I've always followed that thinking. And I think that the MECFS community should too.
Wonderful. Thank you, Dr. Proal. That was an immense amount of information. I learned a lot. I'm still kind of spinning in my head about a lot of misconceptions I had about these interactions of pathogens with our regular bodily functions. So I'll just start really quickly. You mentioned that you regularly post your findings and interesting articles that are in this area in your field on Twitter. So what's your Twitter handle?
It's at microbe-minded2. It's pretty cheesy, but yeah, I really do always think about microbes, so it's fair.
I love it. So is that 2 with the number 2, or
yeah,
is that
2?
Got it. So at micro-minded2.
Microbe-minded.
Microbe-minded too. Got it. I will definitely follow you on Twitter. Wonderful. So we had a lot of questions. They largely fell into 2 areas of specificity, the brain specifically, and then the gut specifically. So with that in mind, you mentioned your Ramsey Award project, your Ramsey Award grant. Could you tell us a little more about your team and what outcomes you're hoping for that Ramsey Award funded grant project?
Sure. So let me go back up to the couple slides I put. Oops. Can I go back up to the other slides?
You should be able to.
Oops. I'm trying.
Oh, no. Okay. Let me see if I can help here.
Okay. Whoops. Yeah. So there's a lot of slides here. I could have talked forever.
That's all right. We'll be happy to post the slides after, folks.
Go up higher. Sorry.
Oh, whoops, wrong way. Wrong way. Sorry.
Yeah.
Okay.
Those were even more slides. Okay. So keep going. And above him. Yes. Okay. So one more up.
There we go. That's the one.
So what we're trying to do is create a pipeline. It's basically a collaboration between 2 or 3 teams. One team is at Berkeley Joint Genome Institute, and they're the team that's doing the Uncovering the Earth's Virome project, which I mentioned is finding many known novel and known viruses in many of Earth's ecosystems. So we want to put all their tools and algorithms to use, but we want to look for organisms in MECFS blood and cerebrospinal fluid. And the reason for that, there's several reasons. Is that, well, the samples are coming from Jonas Bergquist, who's really cool. And what he's going to do is he's going to collect blood and cerebrospinal fluid from the same patients. So it's like, so we can basically look in the blood and find, we're going to look for all kinds of organisms. You can see on this slide, well, that's us, but you can see here, their technology can actually identify, double-stranded RNA viruses and DNA viruses and phages and specific viral groups and retroviruses, as well as bacteria and other organisms in one analysis. So we're not necessarily looking for a single pathogen. We just want to know what organisms are there in the blood with these tools. Step one, right? And we want to know what we can find in the blood, and then we want to know how well that reflects the organisms. We're going to do the same thing to the cerebrospinal fluid from the same patients. And what that allows us to see is like, okay, what's in the blood and what's in the cerebrospinal fluid, and how much of a correlation is there between those organism communities or any, pathogens that we find that seem to be important, right? And one of the reasons for that is because I think there's a misconception out there that if a patient with MECFS has an infection, that organism should be found in the blood, and that really probably isn't the case. Most times when an organism is in the cerebrospinal fluid, or especially in brain tissue, it's not in the blood. And in fact, that is very rare that happens. And so we don't want to keep using blood tests to just the only way to, look for organisms in patients with MECFS if the, infecting agent might be elsewhere. And so, this is such an early topic of research. We don't really know what we're going to find between the blood and the cerebrospinal fluid, but that will at least give us a precedent to know what organisms might be in each space. And beyond that, if we could get MECFS autopsy brains or brain tissue samples, we could even begin to try to see what's in the brain tissue in relation to the cerebrospinal fluid. We don't have autopsy brain yet, but that's a possibility. But what I did to try to address that question is partner this project with a team at Harvard, the ones doing the brain microbiome project, Rob Moyer and Rudy Tanzi. And they're going to run some of their samples through the same pipeline. And they might, with their samples, we could probably better tell what's in Alzheimer's brain tissue, what's in cerebrospinal fluid and what's blood, and what reflects what where, right? So because they have access to brain tissue. So we might bring in even samples from different neuroinflammatory conditions into the whole thing too. It's an ongoing thing. But at this point,
that's fascinating.
MECFS samples will go in that direction. And I want to be clear, this is a first step. This is like what's there. We're not even, well, no, actually part of the algorithms that they have, David and Nikos who are on this, can predict relationships between the organisms in these communities. So we can figure out what phages are infecting what bacteria in the communities, for example, right? We can kind of infer community dynamics, and if those dynamics are dysbiotic or imbalanced, that can be clues as to what pathogens might be driving that dysbiosis. So there's a lot of trends we can find in that. And then, of course, we go off that, and then we can, create new hypotheses for, looking at, other organisms and certain organisms in more specific ways, right? So that's kind of the, that's the basis of the project thus far.
That's really exciting. And actually, I just wanted to say, I'm glad you mentioned the lack of our accessibility to brain tissue samples for people with MECFS, because our organization actually has recognized that is an urgent need in the research space, and we've partnered with a group called the Brain Donor Project, www.braindonorproject.org. And for patients who are interested in donating their body to science after they pass, there is actually a special process that is unique and complicated in order to get brain tissue, and it has to be done with consent while the individual is still alive. So for anyone who's interested in exploring the process to donate your body, specifically your brain tissue, to science after you pass, please visit thebraindonorproject.org, and we've been working with them to facilitate ME samples to researchers who need them. So thank you so much for pointing out that urgent need. It's something that the research space is really looking for.
Awesome. I'm so glad you guys are doing that. And also, just so you know, this sounds like a very well organized thing. In worst case scenario, if you want to donate, you can call the Harvard Brain Bank. It's an 800 number available on the web, and they will come immediately for free to gather samples too. So we could probably talk about that later to make sure we just coordinate. But
Look,
yeah.
a new collaboration just happened right here. I
Yeah.
love it.
But no, that's great. Okay.
Fantastic. So I mentioned earlier that a lot of the questions really focused in on two areas of the body, the brain and the gut. So we'll start with the brain because I personally found that particularly fascinating, especially because Alzheimer's is something that does run in my family along with MECFS. So I really felt a personal connection to that unique relationship, particularly with plaque buildup in the brain.
Yeah.
So one of the first questions we got was relating to the vagus nerve hypothesis. If you're familiar with that theory of what causes MECFS and specifically the blood-brain barrier, could you elaborate about the brain impact of the microbiome and specifically if the brain inflammation that we're seeing could contribute to glaucoma, hearing problems, or even Alzheimer's?
Yes, I mean, so again, this is an ongoing topic of research and it's very new. So there's no, complete conclusions. I don't want to over, emphasize anything, but this is like I mentioned, a big trend in which there are a range of organisms. There's other studies as well, finding organisms in the brains of patients with diseases and even in healthy brains. So if you look at disease the way I do, what I was, I know it was complicated, but when I was going into the way that pathogens and their proteins and metabolites can dysregulate signaling pathways, well, that can happen to so many different kinds of signaling pathways that control all kinds of processes. So you could have a pathogen, if it was in brain tissue, that could probably contribute to any kind of brain dysregulation, related to hearing or this or something that involves the central nervous system via that mechanism. We definitely need a lot more study, but it's certainly possible, right? And then about the
Wow.
vagus nerve, no, it's really important. I know Mike, who is one of my colleagues, Van Elzeker, that's his hypothesis, and I've always thought it made a huge amount of sense. I think though that vagus being infected would result in a lot of symptoms of MECFS, but vagus is a nerve that innervates so many parts of the body. So it communicates to the brain, and then it also, extends into all different body sites, and including the gut. In fact, the gut-brain connection with vagus is very important. It's actually very well studied. It's referred to often as the gut-brain axis. So via the vagus nerve, organisms in the gut can signal to the brain, and theoretically, organisms in the brain can signal to the gut. That hasn't been shown as much, but I mean, it's Robert Moyer, who's on our project, he always describes this as two-way axis. So you're getting communication between organisms in these different body sites as well. So vagus, if it's infected, it could be infected in the gut. It could be infected in the brainstem part. There's different ways that the nerve could become dysregulated that could contribute to symptoms. But also, Mike often talks about what he calls equifinity, which is sort of different, ways, different mechanisms that can result in a similar outcome. It's a little bit like my successive infectious models. Like no one has to have the exact same exposures or infections for a similar disease state to result. Well, vagus, for example, might be infected in some people, but in some people it might be compressed because of a structural issue. Or you might have, vagus could just be, I don't know, there could be intracranial pressure in a different part of the brain. For example, when you asked about blood-brain barrier, and theoretically there is a thick layer of cells that separates the body from the brain. And for a long time, and even in a lot of medical textbooks today, people will say, oh, that barrier keeps organisms from getting into the brain. And there are so many examples now of how organisms can get around that barrier. Number one, if the organism is inside an immune cell, there's a lot of ways it can traffic through that barrier. But there's also, in the last couple of years, there was a discovery that the brain has its own lymphatic system. The lymphatic system, which is lymph, is fluid, like our blood vessels that go throughout our body have lymph vessels next to them, and toxins and stuff from the blood are pushed into the lymph, and the lymph kind of gets them out of the body. So it was thought that this lymph system ended at the neck. But a couple of years ago, this researcher, Jonathan Kipnis, and his team, and actually a second team, found they were just looking at some brain scans, and they were like, what's that? And it turned out that we missed the fact that this lymphatic system does extend into the brain. So what that means is that immune cells can traffic directly through that lymph system into the brain as well. And again, if those cells are infected, that's a total route around the blood-brain barrier. And beyond that, if the lymph, if there's infection in the brain, or blood vessels in the brain are infected, or there's just generally inflammation, that could cause that lymph to swell. So, when you get a cold, your lymph nodes in your neck kind of swell. So imagine the lymph in the brain swelling. That could create intracranial pressure, and that could possibly compress or, impact vagus as well. So you could have,
Wow.
vagus in this disease could be impacted by direct infection. It could be impacted by direct compression, or it could just be kind of dysregulated by other inflammatory changes in the environment as well. So, but yeah, we should very much keep an eye on the vagus nerve, because it is at the very least relaying certain symptoms, I would say, in this disease. It's kind of a signaling, it's an important signaling pathway.
That's fascinating. Actually, so 2 follow-up questions to that point. I'm kind of jumping on them as they come in. So thank you, everyone. Keep your questions coming. The first is, you described kind of once that lymph system is permeated, it can get around the blood-brain barrier, these little phages and
Yeah.
microbes. Is that reversible theoretically? If it's one, is it one of those things that once they're in, and there's nothing you can do about it, or is it potentially reversible?
Yeah, I mean, good question.
We don't know yet is the answer.
Yeah, well, so there's right now we don't know. For example, that's why Rudy Tanzi and that group are trying to do the brain microbiome project. We don't know. Maybe even healthy brains have communities of organisms in them, and maybe that's just okay. Maybe that's, maybe organisms are in the brain, and people, just like they're in the gut, and people can be healthy with that too, right? So I'm not
Wow.
sure if we should be, killing all the organisms in the brain. That's something that totally remains to be discovered, to be seen. But if an organism is a pathogen in the brain, we just don't want it there, right? In terms of getting it out, we're just, treatment is not amazing thus far, but, we could use certain antimicrobials and antivirals and whatever it is. And again, I'm a fan, like I said, of just generally trying to do interventions that would support the immune system itself, because the immune system has evolved so many of it, in simple terms, its own weapons that it can basically use to kill pathogens in any body site. And so if we sort of support the immune system generally, it might be able to, increase the production of cells, or increase the, expression of other antimicrobial peptides that can target pathogens. And so we could kind of help our own immune systems better find these pathogens and kill them. So this is just an ongoing direction of research. And in my opinion, something that should be very prioritized would be the development of, more antivirals, more antimicrobials, more immune supportive strategies, and beyond that, kind of protocols and, better understanding of how they could be used under these different conditions. And yeah, again, that's,
Wonderful. Wow.
early stage.
So the follow-up question to that one comes from a doctor who's on our webinar. And unfortunately, I'm going to read this exactly because I'm not 100% sure about what this means, but I hope it will make sense to you, Dr. Proal.
Okay.
Can external vagus nerve gamma wave 10s be used to address the vagus nerve hypothesis?
Oh man, yeah,
if you could take a moment, because I actually don't understand that question.
Okay, yeah.
If you could explain what that means, I'm sure I'm not the only one, and also I'll answer, of course.
Yeah, so the first thing I want to say is I'm not a medical doctor, so I can't really answer specific questions about treatment because I'm not trained to do that. But generally speaking, so what he's talking about with TENS devices, and there's similar devices that you could basically put like on the nerve, or you can kind of insert something in your ear, or different ways of an external device sort of releasing, Mike Van Elseker should talk about this, but it kind of pulses in a way, sends out a signal that stimulates vagus. And
Oh,
so
okay.
you could have this external device that you put in your ear or on the nerve, depending on the device. And in theory, because it's pulsing in a way that impacts the vagus nerve's activity, it could modulate that. And if the vagus wasn't functioning well, you might be able to sort of up its activity by using one of these devices. And, I think that is a very good, fruitful area of research, and that it would be cool. I think there are some examples of patients with ME/CFS who have used some of these vagus nerve sort of stimulating devices with some success, but it would be cool to study that much more formally.
Absolutely. Well, thank you. I'm just going to try and move us along because we still haven't even touched on the gut yet, and we had a lot of questions there. But would it be okay if I shared your email address with the gentleman who had that question, and then the two of you can go a little bit more in depth there?
Definitely.
Beautiful. Okay, I'll make sure to do that. So now I just wanted to zoom in, if you will, on the gut specifically. We had a lot of questions in that area. So the first one that I'll touch on is, in general, with the literature that you've reviewed and your understanding of these different interactions between these systems in the body, the gut microbiome, specifically fecal transplants, have been an area that have been explored by a lot of patients. Could you take a moment to just describe what you've seen regarding fecal transplants and ME/CFS, and do you know any specifics of any of the outcomes that have been described in the literature so far about fecal transplant process?
So, okay, again, I'm not a doctor, so I can't really talk about specific cases or recommend fecal transplants in any way, but I don't know if I missed it. I'm not sure if there's been a formal trial on fecal transplant in ME/CFS. If there is, I haven't seen it. That being said, I'm sure that there may be individual patients out there who have been trying it. It makes sense in theory as an intervention, possibly. What fecal transplant really is, in simple terms, is that you get a fecal sample, which is just a poo sample, from a healthy person, and then you try to enema those organisms in that sample into your gut so that they could sort of replace what might be an unhealthy or dysbiotic mix of organisms that might be in your gut, right? So if you think that the communities of organisms in your gut are not in a good state and they might be driving symptoms, it's literally a way of trying to get someone else's organisms into you, so that those organisms might, make the community more stable or more healthy again. So you need a donor who's a healthy person who can give you a stool sample that would allow you to do this, right? Now, there's some issues with that. So the FDA, who should be regulating fecal transplants, hasn't really made a call on regulating them. So right now, it's kind of like hard to get one done professionally. There are clinics that kind of do this, and if someone was going to do a fecal transplant, if I would do one in a clinic, because one of the reasons that one of the things I could say is that you're getting a fecal sample or a poo sample from a healthy person, and if you're going to do a fecal transplant, you have to test that sample at least for common pathogens and all kinds of you don't want to assume that someone's healthy only for that person to have some pathogens in their sample that then you are introducing some pathogens into your gut now, right? So the screening of the sample is really important, and that can be done better at a clinic than it can be by an individual person. So what I would like to see with fecal transplants is I would like to see the FDA accept that this is happening, because a lot of people are doing them, and come up with better ways that people could be able to go to a clinic and do this with a professional, as opposed to sometimes people just doing them at home right now by themselves. And as I said, that's, it's a personal choice, I suppose, but you want to make sure that the sample you get from your donor is in good shape.
Thank you so much for that heads up. That does sound like a dangerous self-treatment process.
Yeah, whatever, patients feel really sick, and I get it. But the thing is, again, in ME/CFS, would that improve your symptoms? Well, there's been some studies that have shown some form of gut dysbiosis in patients with ME/CFS. And so maybe, and again, because all of these organisms in the body interact. So when I'm talking about vagus nerve, the communities of organisms in your gut are going to be signaling to the brain, and vice versa. So technically, if you could improve the organisms in your gut, you might improve signaling from the gut to the brain. I mean, it's still not that fleshed out, but there's a possibility that that could benefit patients with ME/CFS, but I would love to see that tested in a more formal trial.
Absolutely. Well, hopefully we'll be able to fund that trial someday.
Yeah.
So continuing with the zoom in on the gut, with the questions we've had there, two common comorbid diagnosis or comorbid conditions that folks with ME/CFS often have in conjunction with their ME/CFS is IBS, irritable bowel syndrome, or episodes of gastroenteritis. I hope I'm pronouncing that correctly. Can you speak to a little bit about how those might be related, given what our conversations about the microbiome specifically in the gut?
Yeah, so I think that they are definitely related, as in, I would say, first of all, so there's a lot of studies that look at patients and they identify some form of gut dysbiosis, and then the conclusion of the study sometimes is like, oh, gut dysbiosis may be, you know, driving this disease or something. One thing you have to, I think about a lot with gut dysbiosis in general is that, okay, for example, there's a study, actually one of my slides showed it, but I could just explain it. HIV, for example, when it infects a patient, as I was saying, it's such an immunosuppressive pathogen. So it knocks down T cell activity. It'll knock down CD8 T cells, CD4 T cells. Those are really important cells that allow the immune system to recognize pathogens. So once a person has an untreated HIV infection, this one team then looked at the gut microbiome environment in patients infected with HIV. And if they weren't treated, then the gut virome especially began to become dysbiotic or imbalanced, because the immune system was no longer able to sort of regulate the pathogens in that community as well. So a lot of these patients with HIV already had enteroviruses in their gut. They had CMV in the gut. They had, I think, I don't know, some herpesviruses in the gut, and those organisms hadn't really been acting up yet. But once the patient was immunocompromised by the HIV, which was basically in the blood, then those viruses in the gut acted up and started shedding more virus particles. And so sometimes my point there is that gut dysbiosis can be a downstream effect of intracellular infection from pathogens in the blood and other body sites. So sometimes when I see gut dysbiosis, and gut dysbiosis has been tied to irritable bowel syndrome and you know, constipation and, you know, diarrhea in many studies. It's, yes, the gut imbalance of gut microbiome communities can drive those symptoms. The question in ME/CFS patient is why. Part of the time I think that if the gut becomes imbalanced like that in a way that can also drive these bowel symptoms, it could possibly be a sign, like with HIV infection, that there's a pathogen in the blood or the brain somewhere also, you know, that's driving a lot of immunosuppression, that's allowing that gut dysbiosis to become a bigger issue and contribute to IBS type symptoms. So to me, it's all connected. To me, it's, I wouldn't, for example, I don't think I mean, a patient with ME/CFS can get a diagnosis of irritable bowel disorder and ME/CFS, but at the end of the day, I think that those symptoms are derived from similar processes.
Wow. Okay. Well, that's really helpful. So we are coming up to the top of our time. So if it's okay, I'm going to try and just do two more questions. There's a whole lot we didn't get to. Would you be maybe willing to, if we sent you a list of questions that we weren't able to get to today, we could answer them by email and put them in a post on our website?
Sure.
Oh, awesome. You're so kind. Dr. Perl, again, thank you so much. You've been so generous with your time and your knowledge. We're so grateful. We've had many compliments about your presentation, including one who said this is the most educational webinar they've seen on ME/CFS, so really positive reception. I'm going to try and just zoom in on the two questions that were asked earlier on in the webinar, and I'm going to try to summarize them as quickly as I can. So really, we did touch on the genetic factor and the fact that some of these things can be just, you know, genetic, they're born with it. So to that end, are there some kind of genetic testing that could be done if we find a contributing factor that could be genetic in nature. And then a follow-up, can CRISPR gene editing be used to discover or correct genome abnormalities that we would potentially see at MECFS?
Yeah, those are good questions. So yes, there are actually companies that can do pretty good human genome kind of analysis to look for variants that can predispose to disease. But one thing I want to talk about with the human genome is I don't personally see evidence thus far that there, and this goes for a lot of diseases, that there is like the MECFS gene. If that were the case, this whole thing would be a lot more clear, right? So I just want to say that, like, to give you some background on that, like around the year 2000, the Human Genome Project was being run, and at that time there was this huge explosion in tools, so people were beginning to sequence our human genomes. And at that point in time, there was so much excitement because a lot of the research community thought for sure that once we just sequenced human genes, we were going to find this simple thing like the Alzheimer's gene, the Parkinson's gene, the MECFS gene, and it was going to be really simple. We'd find that and then we'd kind of fix it and then the person would get better. That has not played out in a single illness community, right? So even in Alzheimer's disease, there's one gene called APOE that can kind of contribute to development of the disease, but only about 20% of patients with Alzheimer's even have that gene. So what the model for genetics that I think most makes sense in MECFS and other related chronic conditions is that patients, okay, at this point in time, you can have your genome sequenced and all the genes that you have what are called variants in them. So you have a gene, but there can be different sort of forms of that gene, and you may have a specific variation of that gene that will allow it to sort of perform its function sort of more one way or the other way in you. So let me give an example of that. Like there are certain gene variants that impact mitochondrial health. So there are, gene variants that can impact already like how well your mitochondria are able to sort of create energy. And depending on what variants you have in that, your mitochondria may or may not be producing energy sort of in a normal level, right? But the thing is with these variants is that they usually require something from the environment to also impact the patient so that the effects of that variant become more of a problem, right? So think about it. Most of us with MECFS or patients who have MECFS, were not born with it, right? So, or some people were, but a lot of cases, people were very healthy and then all of a sudden they got sick or an infection or something happened to them. And that's when the disease started. So what that really infers is that this person could have had a variant that predisposes towards, let's say, impaired mitochondrial function, but that variant wasn't a big enough problem on its own to cause disease yet in that person. They were able to function. But let's say that a person with a gene variant that already predisposes towards mitochondrial problems then gets an infection with a pathogen that also infects and disables mitochondria. Now that's a double hit, right? So now you have the pathogen disabling the mitochondria and that variant pushing the mitochondria in that same direction. That interaction between the gene and the environment exposure is what usually pushes the real symptoms forward. So when I look at the human genome and disease, and I actually work with a guy named Chris Phobes. He has a company named Gene Savvy. I'm not really promoting, I don't want to promote companies on this, but that's the company that I work with when I'm trying to get someone's human genome sequenced, and he's just a really cool colleague and friend of mine. So he can do that. He can take an individual person, go through their genome, come up with variants in the genome that impact that person specifically. Like, your mitochondria kind of have this variant issue. Oh, your immune cell has this issue where, lipid isn't being metabolized so well with your immune cells. And then what I can do with some of that is say, if that patient already has a mitochondrial problem and they had exposure to X pathogen, then that could combine in a way that, that could be kind of a thing, right? So Chris and I are actually working on starting a nonprofit, a different one, where we're going to try to do that, where we're going to increasingly try to look at each MECFS or similar case with that information. So Chris can look at the gene variants that might predispose towards problems, and I'm going to try to better characterize the environmental exposures that can compound those gene-based problems, right? And I think it's the intersection of those different, data sets that can lead to a bigger picture of what might be happening in a specific patient. And so sometimes I'll talk about personalized medicine in MECFS, and that's what I'm really referring to, is like, what things can we find in your genome, and what environmental exposures have you sustained, and how can we fit those together to best explain your case, right? And that's, in a way, if we wanted to, theoretically, would allow for more personalized treatment, because yeah, there are ways that certain drugs can be used to, compensate for human genome variants that are not so great. And there are, of course, like existing antivirals or antimicrobials that could possibly wear down certain pathogens. So if we kind of knew what were the factors in each patient, we could better tailor treatment in theory to that person's specific case, right? Which means that in our minds, it's unlikely that there's going to be like one MECFS drug that you just take that one thing and everyone gets better. It might be a mix of different related interventions tailored for each person.
That's really promising though, and it's really exciting that we're on the path to kind of peeling those layers of the onion.
Yeah.
Okay, so last question, because I know we're already way over time. Thank you everyone for sticking with us. And again, we'll be posting this online, and we'll have follow-up questions with Dr. Proal, because we just overwhelming questions, haven't been able to get to them all. I'm gonna kind of combine a couple questions for our last question. And we've had a couple questions that spoke to stress and physical trauma that impacts people's experience with MECFS. I know that anecdotally, I've met a lot of patients who became sick after a systemic trauma like surgery, or getting sick and having a stressful situation in their life simultaneously. So what could you elaborate on, based on the research and your literature, about the impact of stress on these microbiomes and viromes that live within us? Is that something that impacts their function, and could that be something that exacerbates the biological underpinnings of MECFS?
Yes, definitely. I mean, the most simple way that I would say stress, which is a broad range of, stuff, could be involved, is that a lot of stressful events tend to knock down the immune response, in simple terms, right? So the person's immune system isn't able to function when the body is under, a state of, stress, whatever it is. Sometimes it's pregnancy, sometimes it's this, sometimes it's an accident. And that kind of immune system knockdown associated with that stressful event can allow other factors already contribute, so maybe the person, for example, has an Epstein Barr infection already, but that virus is more in what's called like a latent form. It's in certain cells, but it's not really acting up. It's not expressing its genes in a way where it's really driving symptoms yet. But let's say that person's in an accident, they hit their head or something. Now the immune response goes down, and the virus that's already in their cells can break out, infect more cells, change its gene expression, express more virulence factors. Maybe then it can get into, bypass the blood-brain barrier. Under conditions of stress, there's some studies that show the blood-brain barrier becomes more permeable, so that organisms might be able to get more into the central nervous system. So maybe the stress could allow something in the blood to get into the brain, or maybe stress could allow organisms in the gut to better leak through the gut lining into the blood. So you just have these changes where, in simple terms, when the body's compromised and it's not able to function the way it should, and the immune system is not really able to, when there's an injury, the same kind of cells and parts of the immune system that target pathogens also respond to injury. So injury generates a range of inflammation and problems. So the immune system can get distracted trying to repair parts of the injury, and then it's not turning its attention as well onto the pathogens that might also be around. And so, yeah, again, you're going to get a distraction from that could allow those pathogens to proliferate in a way that after the stressful event, now the person feels more sick because of those other factors accumulating and playing a bigger role.
Wow, I did not realize that stress was that comprehensive of a immune system impact. I mean, that is really something that I think a lot of patients have known anecdotally, but to hear how broad that impact can be is really a little bit terrifying, if I'll be honest.
Yeah. But I want to clarify though that, I think there's sometimes an overemphasis on stress in this condition, in that, sometimes it's assumed first that, oh, person must have been stressed out or something, and that's why they got sick. Well, no, I mean, I don't think that there's more relationship with stress in ME/CFS than there is with anything else, with cancer, with anything. So yeah, it can be a factor, but it's not, I don't think I see a trend in which there's like, oh, ME/CFS is the condition in which stressed out people are more likely to get or something. That's kind of fits back with the psychosomatic narrative that's so ridiculous with this illness that I hate.
I agree with you there. It's definitely frustrating as a caregiver for someone with ME/CFS to have that dismissal with, oh, you're just going through a stressful time.
Yeah.
But I don't want to dismiss the fact that a lot of patients report a lot of improvement when they reduce stress or diet changes or other things that sort of help their body immunologically get better and stronger.
Exactly. And that's consistent across cancers and a lot of stuff. That's a similar trend, but yes, that could be possible for sure.
Wonderful. Well, I'm really excited to see where your Ramsey Award grant project takes you. I'm really optimistic about just all the information you shared is swimming. I'm definitely going to watch this presentation again. So for everyone who joined us, thank you so much. For the questions we didn't get to, we will definitely be following up on those in writing afterwards. So keep an eye on our social media, which you can see on the screen there, Instagram, Twitter, YouTube, and Facebook. Also on our website, www.solveme.org. And I'll just take this last moment as we're concluding, is that these webinars are provided entirely free of charge for everyone in the community. And we are able to do that because of your generous financial support. So now through the end of the year, we have a $750,000 Double Your Impact Challenge. Several generous donors have agreed to match funds up to $750,000 through December 31st. Your donation will literally double your dollar. We're really excited to reach 1.5 million by the end of the year so that we can continue in next year bringing you great programs like this, our advocacy work on Capitol Hill, and of course, our Ramsey Award program and patient registry that allows funding for scientists like Dr. Proal to do their amazing work, and others. If you haven't seen our recent class of Ramsey awardees for 2019, I encourage you to visit our website and check them out. The projects this year are bigger, better than we've seen in previous years, and I'm so thrilled. I'm just exploding with so much excitement for where this field is going and the opportunities that we'll see in the near future. So again, Dr. Proal, thank you so much for your time and joining us. You were more than generous with your time, and we'll look forward to more updates from you and your team soon. Thank you, everyone, and have a wonderful day.
Cool. Thank you. Am I still on? Oh, yeah.